Everyone. It's another hot and steamy day here in New Jersey, so I thought it'd be a good day for some kitchen science. So what we're going to be doing today is we're going to extract DNA from some strawberries. Now, I've posted for you the instructions for the lab. So if you want to follow along at home, or if you want to watch me do it and then do it yourself at a later date, it's a really cool lab and it just uses everyday things that you would have in your kitchen. So you can, do this experiment at home very easily. Okay. So the first thing you're going to need are strawberries. So you can just go to the grocery store, buy a couple of strawberries or buy a package. If you grow them in your backyard, even better. But I'm just going to take a couple out of this container. And what I'm going to do is that first step is to take the green leaves off of them. Okay. So I don't want the leaves in my extraction. They just kind of get clogged up. So we're just looking at the strawberries themselves. One of the reasons we use strawberries is because strawberries are what we call polyploidy, which means they have many different, poly means many, sets of DNA. And because they have so many sets of DNA, when we extract the DNA, it's just a little bit easier for us to see them. Okay, so I'm going to take my strawberries. I'm going to place them into a Ziploc bag. If I can get it open. There we go. Okay. And then I'm going to squish them. Okay. The squishier they are, the better. So just kind of squish them around. Squish, squish, squish. I always make sure I have an extra Ziploc bag just handy. I have one right here on the counter, just in case this one pops so you don't make a mess everywhere. Okay, keep squishing. You don't really want any chunks in there. You want it to look kind of like liquidy, almost like a smoothie, I guess, because you're not going to get it perfectly liquid. You'll have a few little chunks in there. Okay, so now hopefully before you got started, you took your alcohol, your regular rubbing alcohol, and you placed it in the freezer. You want that to be as cold as possible because that's going to help with the extraction of the DNA. We're going to be able to separate it out more easily if it's the colder it is. Also, before you started, hopefully you made up your DNA extraction fluid. It sounds so fancy, doesn't it? So sciencey, but all it is dishwashing soap, a little bit of water, and salt. Okay, so we use the dishwashing soap to break up those lipids in the cell membranes. We're also using the salt because it's a little bit of an abrasive to help break the cell walls, because strawberries are a plant. They're going to have a cell wall, and the water is just to make it a little bit, give us some more liquid. So this is pretty squishy. That's about good. So you can see there's not much solid left in there. Maybe a few pieces. I probably could do a little bit better job, but I'll leave that up to you at home to do a little bit more squishing. All right. So now I'm going to take my bag and open it. And I had previously made up my really fancy DNA extractor liquid in a water bottle. And I'm just going to pour about 10 milliliters into my bag. Now, you don't have to have a graduated cylinder at home, or, even you could do it in a measuring cup if you have one, or you could just kind of eyeball it. Okay, I'm going to put a little bit more. just to make sure it's kind of liquidy. Might add a little more. That was a big strawberry. Okay. Now you're going to zip up your Ziploc bag again and do more squishing. Make sure that you're not making too many bubbles when you squish this time, because if you make too many bubbles, it makes it hard to filter. Okay, so I'm making sure that's really mixed in with my strawberry squish mass here. So if you think about DNA, remember DNA is held in the nucleus of our cells, and it's the same for the strawberry. So that DNA would have been held in the nucleus of each of the cells in that big strawberry that we squished up here. And we should have quite a bit of DNA in this liquid. Okay. I've never tried this with frozen strawberries, but I have a friend that did, and she said it worked just as well. So if you have frozen strawberries in the freezer, you can go ahead and do it with that and let me know how it comes out. I'd love to see your pictures actually, if you do this lab at home. Okay, so now squish with the soap and salt and water. And now what I'm going to do is I'm going to take just a coffee filter. Okay, your parents probably have them in the kitchen somewhere. It doesn't matter what shape it is or what color. I like the ones that aren't bleached, so I buy the brown ones. But if you have the white ones, those work just as well. And you're going to stick it in some kind of a clear container. You can use a jar like I did. I had this jar on my counter, so I figured I'd just use that. You have a drinking glass that's clear, you can use that as well. Okay, anything you have is fine. What you're going to do next is you're going to pour this liquid into the filter paper. Don't stir it up, don't stick a pencil in there or anything, because if you break the paper, the whole process is going to take you a lot longer. Okay, so I poured my liquid into the top. Okay, I don't think I can really, oh yeah, I can show you. There you go. So it's sitting there in the top. And now what'll happen, and it's starting already, is as the paper becomes saturated, the liquid will filter out into the container, and I'll be able to extract the DNA from that. So what's happening is the solid part of the strawberry that was left, even after I squished it, it's going to get trapped in that filter paper. The liquid, that's going to include some of that soap, a little bit of the water. It's going to come through the filter paper slowly, collect it in the bottom, and then I can extract my DNA from that liquid. Remember, DNA is a tiny, tiny molecule. We can't see one strand of DNA with our naked eye at all. The only reason we're going to be able to see this is because it will be lots of DNA. I'm going to pause the video here so that this can drip through because it takes a little while, and then I'll pick it up as soon as I have enough for us to extract. Okay, see you in a little bit.